A good morning or afternoon to you. Thank you for joining us. I'm Jan Reichert, Chief Operating Officer of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members, as well as the broader scientific community, about topics relating to antibody discovery and development. Our expert speaker, Dr. Jin Lu, will discuss how GS discovery and bYlok technologies can help advance bispecific discovery programs towards clinical success. Please note the webinar is being recorded. Please do add any of those questions in the Q&A box in the viewer, and they will be answered at the end of the webinar. Without further ado, I'll now turn the show over to our speaker.
Thanks, Janice. Hello, everyone. My name is Jin. I'm the Senior Technical Support Manager for the Lonza licensing team. Today, it's my pleasure to be here to present Lonza technology. Today, I will be presenting two of our advanced early-stage bispecific discovery programs, GS Discovery and bYlok bispecific pairing technology. Before we begin, I would like to give a brief introduction to Lonza Biologics. We have industry-leading expertise in cell construction, process development, as well as manufacturing drug substance for both monoclonal antibodies and other recombinant proteins. Additionally, we developed and manufacture bioconjugates. In recent years, we have also established a leading position in mRNA drug substance production. We offer a wide range of expression technologies and tools to meet various needs and achieve these goals. Our expression technologies include GS gene expression system, GS effect cell line, GS discovery transient expression platform, bYlok bispecific engineering technology, and XS technologies. At Lonza, we use our GS gene expression system to produce a wide range of molecule types, including standard monoclonal antibodies and other molecule formats. As you can see from this slide, the GS system has demonstrated its effectiveness across diverse molecule types, and we use it extensively for all our cell line construction and service work. At Lonza, we offer an integrated GS gene expression system, making your expression process easy from discovery through to commercial production. In the GS system, we provide a host cell, vector, innovative technologies, and extensive know-how along with our technical support throughout your product journey. Last year, we launched several new offerings, highlighted in. We introduced our GS effect cell line for developing antibodies with enhanced potency. Additionally, we have our own ThoroPro media and feed system version 9.6. Last year, we also launched the GS Discovery Transient Expression System. It is an excellent platform for generating small quantities of product in a short time span. I will explain how this platform works and how it can benefit you later in this presentation. One of the technologies that Lonza applies is the GS piggyback technology. which help boost product yield and specific productivity. This technology was launched in 2019. Here is how it works. The system consists of two parts: a plasmid containing your gene of interest and the GS-piggyback transposase. The system operates using a cut and paste mechanism. Your plasmid contains the gene of interest and an inverted terminal repeat sequence, ITR. You transfer both the plasmid and transposase into the cell. The transposase recognize the ITR sequences, cut and paste them into open regions of the genome associated with highly expressed genes. G-squared. G-squared vectors are one of the key components of our GS system. G-squared system enable you to simultaneously express between 1 and 4 product genes from one GS vector. This vector contains the GS piggyback ITR sequences, so it's compatible with our piggyback technology for creating high productivity pools and clones. In the GS piggyback G-squared vector system, you will have path vectors together with destination vectors. You insert the gene of interest into the part vectors using standard cut and paste cloning method. Once you have assembled your gene of interest into the part vectors, you use the golden gate assembly method to transfer them into the destination vectors. We have expression vectors at Lonza optimized for single, double, triple, and quadruple gene expression. So in summary, this system is a two-step method. Step 1 involves cloning the gene cassettes into part vectors, and step 2 involves assembling the final GS piggyback vectors. In terms of the titer you can expect from using the Lonza GS system, I have some data displayed on the graph here. This data collected since 2020 from our various global manufacturing sites shows the titer obtained from the lead clone at the early stage in the cell line construction process. The data covers a wide range of different product types that we have produced at Lonza since 2020. As you can see the data here, the harvest titers can go up to 9 grams per liter, and the middle harvest titer using our GS piggyback technology and the latest media is 5 grams per liter across a range of formats and complex proteins. All technologies I have introduced today can be obtained through licensing agreements with Lonza. We offer 2 forms of licensing: research licenses and commercial licenses, which covers preclinical work and commercial use of the technologies. In the next few slides, I would like to introduce you to our GS Discovery platform, a hybrid approach for optimizing transient protein expression. To support large-scale manufacturing, you need to generate a clone cell line, which is resource-intensive and lengthy process. Meanwhile, on the other end of gene expression technologies is transient gene expression, which is typically used to accelerate material supply for early-stage drug development. If you place clone cell line construction on one and transient expression on the other end. Stable pools would sit in the middle. Stable pools have been widely used in recent years, including at Lonza, where we extensively use stable pool for process optimization. When preparing stable pools, the workflow involves preparing DNA vectors, followed by transfection. Then you proceed to the selection and expansion stage of stable pools, where you use specific media, add selection reagents to selectively grow and grow the well-transfected cells. Next, you enter the production stage supported by specific media and phase to give you high harvest titer. Now, let's compare this process with traditional transient methods. You would need to prepare the same vectors, then proceed with transfection. Although you may vary your transfection process here, you might use chemical transfection instead of electroporation for stable pool. The selection and expansion stages are removed, and you go straight into your production stage. Comparing transient to stable pool process, transient platforms are faster and lower in cost. However, you will experience a reduction in titer as less resource is invested. As I mentioned earlier, at Lonza, we use the GS piggyback technology for a significant amount of stable expression work, and we have made many improvements in this area. Therefore, we wanted to apply the experience we gained from stable pools to the transient expression workflow. We have developed a new transient expression platform called GS Discovery, based on our GS piggyback transposase technology. In this platform, we prepare the DNA using our piggyback compatible vector, G-squared vector, as I mentioned earlier. We then perform chemical transfection, simultaneously transfecting the GS piggyback vector and the GS piggyback transposase. This allows the process into a hybrid production stage. During this stage, we can apply low-level selection to the culture post-transfection. We then expand the cells, leading to a harvest after 14 days. The involvement of the piggyback technology significantly increased the titer. To demonstrate this point, I have displayed some data on the graph here for a range of different products. Monoclonal antibodies are shown on the left, bispecific molecules in the middle, and bispecific antibodies engineered using bYlok technology on the right. I will discuss bYlok technology later in the presentation in a few slides. The titer obtained from our new GS piggyback transient process are shown in dark blue. The titers obtained from the legacy electroporation-based transient system are shown in light blue. As you can see, the new GS piggyback transient process yields a substantial increase in titers, up to 30-fold in some cases. We have also included titers from a well-known third-party system on this slide. you can see that the GS discovery platform is either exceeding or matching the performance of that system, marking a significant improvement in Lonza's transient expression performance. Now, of course, high titers means nothing if you don't have a sufficient product quality. To investigate this, we took the IgG monoclonal antibodies material shown on the previous slide and compared the product quality attributes of the material generated from GS discovery transient process to those from clonal material produced using GS piggyback. The slide here shows the main isoforms, major unlinked glycans, and the aggregation levels. The key takeaway message is that because we use the same host cell line, the same expression vectors, same GS piggyback platform, we are able to achieve similar CHO-like product quality attributes for this product in both GS discovery transient expression process and GS stable pool expression platform. Lastly, I would like to introduce some new tools we have added to our GS discovery toolbox. While we introduce the GS piggyback transient process, we understand that a 14-day time frame may be too long for some of our customers and may not suit everyone's needs. The GS piggyback transient process is recommended for maximizing overall product yield, but it does require a longer culture duration. So we have introduced a standard non-piggyback protocol to provide options based on your priorities and requirement. On this slide, we have compared standard non-piggyback transient and piggyback transient processes for 3 different products, 2 monoclonal antibodies on the left and in the middle, and bYlok bispecific antibodies on the right. The standard transient process is shown in light blue, and the GS piggyback transient process is shown in dark blue. As you can see, the GS piggyback process shows a significant increase in titer between day 10 and day 14. However, until about day 10, the titers between 2 processes are similar. This means that if speed is your priority, you can choose the standard non-piggyback process, which is simpler and still have a decent titers. But if maximizing titer is your priority, investing more resources can result in a significant yield boost between day 10 and day 14. This is to maximize product titer and support your development work. Before we conclude this section, I would like to mention that at Lonza, we offer an optimized set of expression tools to support high titers at every stage. All the technologies I have discussed in this section, including GS discovery, GS piggyback, G-scored vectors, and associated protocols, are available to use in your lab through our licensing agreements. In the next section, I would like to discuss Lonza's bYlok bispecific pairing technology. This technology is designed to address heavy light chain mispairing in bispecific antibodies. To set the scene, the number of bispecific molecules entering the clinic is rapidly growing. Since 2011, over 400 bispecific molecules have successfully entered clinical trials. In 2023 alone, we saw the introduction of 120 new molecules. The benefits of bispecifics are pretty clear, and the trend of the bispecific molecules development is increasing. Despite their advantages, generating bispecific antibodies can be challenging. For example, as I show here, in the classic 4-chain bispecific IgG, instead of correctly assembled product with blue and green chains pairing nicely, incorrect pairing can occur, where blue chains pair with blue and green chains pair with green. This can reduce the yield of correctly assembled product and increase the cost of goods. However, this challenge has been effectively addressed by the knob into hole technology. This method introduces mutations to the CH3 domains of the heavy chains, creating a knob and hole structure. This innovation is great for driving the correct pairing of heavy chain to heavy chain of bispecific antibody production. Now, it is considered as the gold standard and a well-known technology in the industry when designing bispecific antibodies. However, even if you have a correctly assembled heavy chain products, you can still encounter heavy chain, light chain mispairing during bispecific production. Some examples are shown here. Again, this will reduce the yield of correctly assembled products, so manufacturing such bispecifics remains a challenge. Lonza has developed a new technology, bYlok, to tackle this particular challenge, which involves some engineering steps to the antibody. The bYlok technology involves elimination of the native disulfide bond between the heavy and light chain in the CH1Cl domains in one Fab arm, and the generation of an artificial disulfide bond between cysteines at the interface of the VH and VL domains. These minor modifications favors the formation of correct heavy chain, light chain pairing. When using this technology together with knob into hole, we can increase the production of correctly assembled products, where the heavy chain and light chain are assembled correctly. I will demonstrate this in the next few slides. Let me explain how we apply this technology. Here we have two standard antibodies. When we combine these two to create a bispecific, we can have a few combinations using knob hole and bYlok technology together. For all the cases I refer to here, we applied bYlok together with knob hole. However, there's no reason why bYlok couldn't work with other heavy chain pairing solutions. As you can see here, 4 different combinations can be generated. We put mutation on one of the Fab arm on the same side as the hole, but you also can introduce the mutations to the Fab arms on the same side as knob. You even can apply it to the other orientation. Typically, When using this technology, we test 4 different versions and find out which one works the best for your molecule. Now I will run through some experimental case studies we generated internally at Lonza to demonstrate the benefits of this technology. In this case study, we selected some monoclonal antibodies to generate bispecific antibodies and clones them into expression vector. Then we express them in a control cell line at different scales. We use a 150 ml scale and also scaled it up to 5 liter. The titer and the product recovery at both small and large scales were evaluated, and we also performed functional characterization of the expressed biospecific antibodies. In terms of the molecules selected for this case study, we constructed them using some well-known antibodies. Here we have trastuzumab targeting HER2, panitumumab targeting EGFR, and alemtuzumab targeting CD52, colored in blue, pink, and green, respectively. On the left side, we generated these molecules in 4 different versions. We have KIH3 and 10. They are the molecules without bYlok technology applied, serving as a knob into hole control. We also have bYlok 3 and 10. They are the molecules with combined bYlok and knob into hole approach, but mutations introduced at different chains. On the right side of the screen, we apply the same approach, combining green with pink to generate 4 different molecules, 2 bYlok and 2 knob into hole controls. Next, we wanted to characterize expressed antibodies, and we did this by following the workflow below. So we identified the correct assembly ratio using LC-MS. We also assessed product quality. Furthermore, we employed a range of analytical techniques to evaluate specificity and affinity. Additionally, we analyzed the stability and immunogenicity of these molecules. I will explain the details in the next few slides. This slide demonstrates the correct assembly ratio for bispecifics, with bYloks shown in dark blue and knob hole control in light blue. For both small scale and 5 liter scale, the results are consistent. For the knob into hole control, we observed between 30 to 35% mispairing product. Bispecific molecules generated with bYlok consistently display greater than 95% correct heavy light chain pairing. We then move on to conduct our functional assay, focusing on the bYlok 10 molecule. We used an ELISA method based on biotin and streptavidin to assess the binding specificity of the bispecific antibodies. Antigen 1, shown in blue, was immobilized on the plate, while antigen 2, shown in green, was conjugated to biotin but not immobilized. We use HRP conjugated streptavidin to indicate antibody binding. As demonstrated by the data presented here, there is a clear correlation between the antibody concentration and its absorbance. This demonstrates that bispecifics generated using Lonza's bYlok technology retain the antigen binding properties of the input parental monoclonal antibodies. Now we wanted to gain a deeper understanding of the affinity of the molecules. Once again, we focus on the molecule bYlok 10 here. We compare the binding kinetics of both bYlok 10 and the knob into whole 10 control. We also tested both parental monoclonal antibodies. From the KD values obtained here, the key takeaway message is that the biospecific antibody generated using bYlok shows comparable affinity and binding kinetics to knob into whole only controls. We also tested the thermal stability using nano DSF for all molecules listed. The result in this slide indicate that the thermal stability of the bYlok molecules is similar to that of the knob into whole only controls. Immunogenicity is a critical factor to consider when assessing the clinical efficacy of any antibody. Here we are employing the epibase in silico HLA class II binding cell epitope profiling platform to evaluate the risk. Essentially, this method produces a DRB1 score. This score provides a measure of how likely the antibody is to induce an immune response in the body. We compared all 4 molecules and broke down the scores into individual assessment of each light chain, knob hole heavy chains, and full antibodies. The data shows the bYlok modifications have a little to no contribution to the overall scores of the different chains. The addition of the bYlok to the knob into whole does not increase predicted immunogenicity. To assess the feasibility of downstream process, we purify the antibodies using Lonza's standard downstream process workflow. We then analyze the purified bispecific antibodies, focusing on standard metrics such as aggregates, fragments, and HCPs. The key takeaway message here is that the antibodies generated with bYlok technology can be effectively purified using standard downstream process. In the next few slides, I would like to show you how bYlok compares to other pairing solutions currently on the market. We compared the bYlok technology to the other 2 pairing solutions currently on the market. They all involve engineering the disulfides. Here we have DM1, which involves mutation to the light chain at the constate region, and DM2, which involves a more significant change to the sequences. The results from transient expression, stable pool, and scaled-down cell line construction were evaluated. Four combinations were tested using transient expression. The best formats were chosen based on transient expression readout, including the percentage of correct pairing, functionality, aggregation, and impurities. Once the best format was chosen, their stable pool expression was also evaluated using GS-piggyback transposase technology. The product was produced in a scaled-down fed batch bioreactor. The results show that bYlok has a comparable stable pool titer to other technologies. Then we proceed with a head-to-head evaluation of cell line construction. An established cell line construction workflow was used to test them head-to-head. The workflow is as follows. We use the Beacon platform for our cloning process and assessed clone productivity. Clones were selected based on Beacon on-chip productivity. Clones were ranked based on product concentration on day 4 in the shake flask. Product concentration, QP, and metabolite were analyzed. The top 6 clones were selected for the product quality analysis. This slide shows the product assembly ratios for the top 6 clones by bYlok and DM1 technology. The method we used here is native seq-MS. It is clear that the product assembly using DM1 technology shows significant variation among the top 6 clones. However, the top 6 clones expressing the bYlok scaffold demonstrate more consistent full product assembly. These slides present the comparison result between bYlok and DM2 technology. Once again, it demonstrates that among the top 6 clones, the bYlok scaffold shows more consistent full product assembly compared to the established domain modification alternative. This slide shows the expression results of the lead clone. So the lead clone was selected based on correct assembly, functionality, titer, aggregation, and impurities. From the expression of the selected lead clone, bYlok demonstrated consistent performance across all key metrics of titer, assembly, and purity. A short summary of this part. The bYlok technology provides an easy-to-implement solution for heavy chain, light chain mispairing and for the manufacturing of IgG-like bispecific antibodies. It has a minimal modification to the natural monoclonal antibody design and fits into standard manufacturing workflows. As I mentioned, it can generate more than 95% correct heavy chain, light chain pairing. It is a portable and versatile platform. Data shown today demonstrated that immunogenicity, thermal stability, and the binding properties of the bispecific antibodies are not affected. Furthermore, combined with our GS discovery and GS piggyback technology, we can even help boost bYlok bispecific antibody titers in both transient and stable pool formats. For the last few slides today, I would like to show you some data we generated to showcase how you can boost bYlok bispecific antibody expression with the GS toolbox. The slide shows the result from our GS discovery transient expression of bYlok bispecific antibodies. Like I mentioned in the presentation earlier, GS discovery is designed for generating small amounts of material in a short time span. With the piggyback technology, you combine both pool selection and expansion at same stage and harvest product at day 14. The right side of the slide shows the comparison result between Lonza's GS discovery transient expression and the legacy electroporation process for two bYlok products. We achieved a 24-fold and 12-fold increase respectively. The GS discovery transient system offers a newly optimized transient process built around GS piggyback that achieves a big titer increase of bYlok molecules compared to the legacy transient process. This slide demonstrates the titer results of stable pool expression using GS piggyback compared to random integration for 3 different products, including bYlok molecules. GS piggyback stable pools expressing bYlok molecules generate higher titers than random integration controls. Thank you very much. That's the last slide of my presentation.
Excellent, excellent. Thank you so much. We have a number of questions. I've tried to put them or group them into categories, with my initial category being the more general of the questions. So I'll start with those. First question is then, is the vector used for stable pool generation the same as the one used for GS discovery transient transfections? And with a follow-up question on it, is the piggyback enzyme used for the stable cell line development the same as the one used for GS discovery transient transfection?
Thanks, Janice. Thanks, everyone. That's a great question. So same vectors is used for both to offer extra flexibility for your development work. For the second following-up question, for stable pools, mRNA piggyback is used, and for transient transfection for our GS discovery offering, the piggyback is used as PTG-23 plasmid. We would like the GS discovery to be as robust as possible, so plasmid PTGE23 is used as it's easier to handle and it's easier for you to use in your lab.
Thanks very much. Next question. Well, it did seem like you were referring to the bYlok as being versatile. And so the question is, what types of molecules can bYlok be applied to?
So bYlok can be applied to classic 4-chain IgG-like bispecific molecules. That means your bispecific molecules consist of 2 different heavy chains and 2 different light chains. That's the application of our bYlok technology.
And follow on with that refers to or asks about the expression level. Does bYlok affect the expression level of the molecules?
So as you can see from the data I presented today, we demonstrated that the bYlok has comparable titer compared to the knob into hole controls, so it doesn't impact.
Very good. Now, a very general question. What impact does bYlok have on overall bispecific antibody development, their timelines, that is, as well as cost?
Thank you very much. Again, it's a really good question. So bYlok can de-risk the clone selection process as clones shows more consistent product assembly compared to the competing technologies on the market. It can also increase the correctly assembled product ratio and assist with the downstream process. So it's actually overall, it won't increase the timeline and the cost in long run.
And now we have a few more specific questions. Given that the expression of the protein drug is transient in nature, how stable is the typical transient cell line? With follow-up question, are transient cell lines used for manufacturing processes that utilize rolling inoculum stages that can go up to 100 plus days of passaging from cell bank thaw?
If I understand, okay, so it's again really good question. For our GS discovery transient expression, we typically use it for the early stage drug discovery for create the material for rapidly create some small amount of molecule product. However, we don't recommend using it for the cell line generation. So if you would like to for the cell line creation, then we would recommend to switch to our stable pool expression technology, also as part of our GS toolbox.
The following question came in relatively early on in the presentation, as to orient you as to what content you were talking about at the time. And the question is, what about specific titer ranges for bispecifics?
So the specific titer range. So one of the data I shown here, I think we can go above about. That's a really good question. I actually have it in my slides. Let me have a look. I think it's about the one in my slide. Can I get back to that question? I just
Sure.
let me quickly. Yeah, if you let me quickly go. If you can ask the next question, I'll get back to you that question.
Hey, marvelous multitasking going on there. Sure, I can ask the next question, which is fairly specific. What mutations are used to eliminate the CH1Cl disulfide in the bYlok technology?
Sorry, because I shouldn't do that. I should have done multitasking. Sorry, what's your question?
I was super impressed because I cannot do that myself. Do you want to find the slide first or answer the question?
No, can you just repeat the question?
Sure. No problem. What mutations are used to eliminate the CH1Cl disulfide in the bYlok technology?
So the mutation, yes. So I can't actually remember exact the numbers. So we made 4 mutations. So in order to create 2 disulfide, so it's going to be 4. And then if you like the details, what exactly they change, and 4 mutations, I will be able to provide them to you. So if you go with Lonza bYlok platform, we will give you a kind of a software. So then what you do is you put your sequence in, and then the platform will tell you which 4 mutation you need to make and what kind of, as I mentioned, it's going to have 4 different combinations, and then they're going to recommend to you, and you will have all those information in our platform.
Very good. Next question, I'm not certain is referring to the same mutations, but you'll decide that because you're the expert. The question came in a few minutes after the one you just answered, and it is, can the mutations be applied to lambda light chains?
So we don't say, we believe as long as your bispecific is 2 different heavy chains, 2 different light chains, there's no reason why it doesn't work for the lambda. So that should work, yeah.
Okay. Next question, do expression variabilities, for example, between 4 different chains in bispecific antibodies limit the titer, even if the pairing is theoretically perfect?
If I understand it correctly, you meant, will the bYlok again impact the titer because we made a mutation? As I mentioned previously, we don't think it's going to impact the titer because we do have the data demonstrate our molecules using the bYlok and knob into hole has comparable titer to the knob into hole only control. So then that won't impact the titer. But I would say here the only thing you need to do is all the same technology on the market. They all do the same. You will have to screen which combination, which mutate combination would help, would work best. So you will have to do a pre-screening work and pick which going to work for your molecule.
And the last question, are there already examples of bispecifics? Whoopsie, I lied, another one came in, but I will finish the one I started. Are there already examples of bispecifics with bYlok format in advanced preclinical or initial clinical testing?
As I mentioned, this technology actually we only launched last year. However, we do have more and more customers signed agreement with us. Although our technology is new on the market, it's only last year, but however, currently on the market, there are a lot of other technology, similar pairing solutions already for a long time. So that's why we're confident that you could using this mutation or disulfide, the technologies, yes.
And the next question, which may be the last one. Oh, no, somebody did it again. There's another question. All right. Is there special requirements on the cell culture medium and condition with the bYlok molecule?
No, we don't. So we no, then it's all fit into the standard media feed, standard downstream process. Everything is standard. Yes.
Okay, and maybe this is the last question. Does the molecule orientation, or knobs versus hole, affect heterodimerization efficiency?
So the only thing, as I mentioned here, would be because you could have 4 different combinations, so you will need to pick the best one. So you will have to do a pre-screen work. But as long as you pick the best one, we believe it will work, and you will be able to generate at least Lonza's internal data. We are able to generate more than 95% of correctly assembled product.
And the next question, would Lonza be exclusively promoting the piggyback technology as part of your CLC service, or is electroporation still an option?
I'm not sure if I understand this question, but I will try my best to answer. So yes, Lonza offer our CDMO service. We offer GS piggyback as our standard offering. And then when you do the transfection so we do electroporation for the stable pool. But for the GS discovery, we switch from the electroporation to the chemical transfection. The reason is for GS discovery, we would like it to be as robust, as simple, as easy possible. That's why we switch from the electroporation to the chemical transfection. But for the piggyback, the stable pool offering is still the electroporation.
All right. Well, I think we're at the end of the questions with the one outstanding one. If you have 1 minute that you could take, if you want, to try to find that slide you were looking for to address the question about specific titer ranges for bispecifics.
So I do have a bispecific, but however, the currently data I have is actually let me see, is actually the it's the transient. Sorry, it's only the transient GS discovery. So with the GS discovery offering the transient expression for the bYlok, we have the internal data prove that we have almost like 360 mg per ml. That's what we got for the bYlok.
Excellent. Excellent. Just double check once more. Last chance, gone, or asking questions. Thanks very much to the audience for all those great questions. In concluding, I'd like to thank Dr. Lu for providing insights into how GS discovery and bYlok technologies can be used to advance bispecific discovery programs towards clinical success. And I thank you very much for joining the webinar today. An on-demand version will be available very soon. I'll send a link to it by email to everyone who registered. Please do feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again and have a great rest of your day.
Thanks, everyone. Thank you very much.